First, we discuss about the definition of the actinomycetes. So, actinomyces are a gram-positive filamentous non-acid fast anaerobic to microphilic bacteria that typically colonize the human mouth, urogenital tract and gastrointestinal tract, but can causes as infection is known as actinomycosis. Different natural sources of actinomycetes from soil, from water, from root and stem sample. First, from soil. Actinomycetes are a group of bacteria that are commonly found in soil and are known for their role in breaking down organic materials and improving soil health. Function. Actinomycetes are important for soil health because they decompose organic materials. They break down resistant organic materials like cellulose and chitin, improving soil structure. They help bind soil together and form stable humus, improving nutrient availability. They help make nutrients and minerals more available to plant. Promote plant growth. They help plant grow by facilitating the assimilation of nutrients and minerals. Control pests and diseases. They can be used as biological controls to fight plant diseases and insect pests. Produce antibiotics and other substances. They are a source of antibiotics, enzymes, and other biologically active substances. Second, from water. Actinomycetes are a complex group of bacteria present in a wide variety of environments, either as dormant spores or actively growing. Some actinomycetes produce two potent terpenoids, geosmin and 2-methyl isobornol, and pyrazine, common cause of drinking water of flavors, and have been implicated in taste and odor episodes. However, isolation from a water source is not evidence that actinomycetes caused a taste and odor event. Dormant spores of actinomycetes may be isolated from aquatic environments in high concentration. Despite production in the terrestrial environment, similarly, odorous compounds produced by actinomycetes may be produced terrestrial and washed into aquatic environments, with or without the actinomycetes that produce them. Actinomycetes may exist as actively growing mycelium in small, specialized habits within an aquatic system, but their odorous compounds may influence a wider area. Actinomycetes can be isolated from water sample using the spread plate technique on starch casein agar. First, take a 1 ml of aliquot of the water sample. Next, spread the sample evenly on a sterilized starch casein agar plate using a L-shaped glass rod. Incubate the plate at 13 degrees Celsius from 7 to 14 days. Next, observe the plate periodically for the growth of actinomycete. Then, select pure colonies, isolate them, and maintain them in starch casein agar slants at 4. Third, from root and stem sample of the plant. Source: Actinomycetes. Isolation of actinomycetes from root and stem sample of the plant such as Cinnamomum zeylanicum, Zingiber spectabilia, Elyterospis cortesii, Libisa pumila. For media, starch yeast casein agar, actinomycetes isolation agar, humic acid, vitamin Gillian gum, tap water, yeast extract agar, coil vitamin agar. Life cycle of the actinomycetes. First, spore germination. Second, mycelia. Then, maturation, reproductive hyphae, Spectation and free spore. Septation and free spores. Applications of the actinomycetes. Actinomycetes are a group of bacteria that have many applications in agriculture, medicine, and the environment. Agriculture. Actinomycetes are used to control plant diseases and insect pests and to promote plant growth. They can produce hypohormones that help a plant grow by increasing the surface area of their roots. Actinomycetes can also fix nitrogen biologically and solubilize phosphate. Medicine. Actinomycetes are a major source of antibiotics, including antibacterial, antifungal, anti-inflammatory, and anti-cancer drugs. Environment. Actinomycetes are important in soil ecology, where they produce enzymes that break down organic plant material, lignin and chitin. This helps from compost. Actinomycetes can also degrade plastics and polymers. Fruit. Actinomycetes are used in the fruit industry. Biotechnology. Actinomycetes are used to produce enzymes such as protease, cellulase, chitase, amylase, and xylase. These enzymes are used in biotechnological industries and biomedical